Hello, I'm going to be presenting on the Global Health Institute Scholars Program that I participated in the summer of 2024. My name is Priscilla, and I'm a second-year PhD student in the Environmental Health Sciences program, and this project that I will be talking about today is being done in collaboration with the Universidad Autónoma de Tamaulipas in Mexico. The research site is in the state of Tamaulipas in a community called La Pesca that is located along the coast of the Gulf of Mexico. Here is a close-up of the research site with all 18 sampling sites that I sampled this summer. In the community of La Pesca, residents rely on shallow groundwater as their primary source of household water and have a high dietary reliance on local seafood and poultry grown in or near homes. As a result, the community is routinely exposed to chemicals from local industry, pesticides from agricultural runoff, and pathogens from untreated sewage discharges. People living in La Pesca face multiple challenges, including a lack of safe drinking water, no wastewater infrastructures, a high dietary reliance on seafood and oysters grown in contaminated waters, cohabitation with domestic and wildlife animals, and overall declining ecological conditions due to climate change. The title of the project is Evaluation of Pathogens, Chemicals, and Heavy Metals in Human, Animal, and Aquatic Environments in La Pesca, and overall encompasses three specific aims, but I will focus on the first aim today, which is to quantify the temporal and spatial patterns of pathogens and heavy metals in water, sediment, and aquatic fauna. The research activities that were completed over the summer included collecting surface water and sediment samples from 18 different sites. In addition, in collaboration with Universidad Autónoma de Tamaulipas, aquatic fauna samples were collected at 6 sites. This included 2 fish samples, 2 oyster samples, and 2 shrimp samples. Physiochemical parameters were also taken at each site. Overall, I want to accomplish building a comprehensive One Health approach that utilizes environmental factors, microbiology and water quality, and human exposures. I am currently working on analyzing all the pathogen data, but here are the preliminary results from the physiochemical parameters that were taken. Parameters included water temperature, pH, salinity, turbidity, and dissolved oxygen. Parameters like pH are important because low pH in water can increase the solubility of heavy metals like lead, copper, and zinc, making them more likely to contaminate the water and harm both human health and the environment. Low dissolved oxygen levels can indicate high levels of organic pollution such as sewage, agricultural runoff, or decaying plant matter. And turbidity can facilitate the transport of pathogens such as bacteria and viruses and chemical pollutants that attach to particles in the water. My main takeaway from participating in this research is that fieldwork is very important, can be hard, but very rewarding, and accomplishing this research has allowed me to gain invaluable experience in global health and One Health, which include practicing my Spanish, improving my sampling methodology, both in the lab and in the field, and engaging with the community. It was nice to put faces and places to the data that I collect. That concludes my summary for my GHI experience. Thank you very much for listening.